This study presents a physical model that connects cell wall microstructure and microfibril motions to complex stress-strain behaviors of extensible plant cell walls. As background, for many years plant biochemists and cell biologists have characterized the polymers that make up the plant cell wall and speculated on how they are connected to each other by strong and weak bonds. Other researchers measured the mechanical properties of cell walls, properties such as elasticity and plastic deformation, without understanding the exact molecular basis of these physical properties. In this study, we synthesized these two approaches to draw insights into the molecular basis of cell wall structure and mechanics. Our model is comprised of three wall polymers: cellulose, shown here as purple chains, xyloglucan, shown as green chains, and pectins, shown as yellow chains. These different chains in the model were endowed with physical properties based upon the actual polymers and allowed to reorganize themselves into a low-energy state to assemble a cell wall. This image shows an example of such a structure. You can see that the three classes of polymers arrange themselves in distinctive forms. We then stretched this digital wall and measured the forces that developed as the wall resisted stretching. The following video clip shows how these polymers dynamically reorganize themselves as the cell wall is stretched. This video will play twice. We start with a square piece of the cell wall which is stretched horizontally. You can see how the cellulose microfibril network gets rearranged by changes in cellulose bundling as well as by sliding of cellulose microfibrils. Now we go backwards in time. It's just for fun, but it gives a different and somewhat bizarre view of wall polymer dynamics. You can see xyloglucan and pectin chains and how they are dynamically rearranged. Next, we'll go forward in time once again so you can see how cellulose microfibrils become straighter as the wall stretches. Their bundling changes and they slide a bit like the way an extension ladder works. Likewise, xyloglucans and pectins are rearranged. Here are two of the key insights from this study. First, cellulose microfibrils form a strong covalent network that transmits in-plane tensile forces in the cell wall and determines stress-dependent elasticity, stiffening, and plasticity beyond a yield threshold. Second, wall plasticity originates from fibril-fibril sliding in the cellulose network. This work was funded by the Department of Energy through the Center for Lignocellulose Structure and Formation and Energy Frontiers Research Center.